Hello. We are the Cellular Brothers. We make cool science videos with bad jokes, showing little children like you how to science. Today, we will be teaching you how to DNA.
In today's video, we will show you how to DNA. We will start off with DNA structure and function. You may have seen this very cool matrix thingy.
Well, today we are going to teach you all about it. It may look very complex, but we will break it down to a simple form. You can try following along with us by opening your standard issue biology textbook. You may wonder who found out how this looks. There were two dudes named James Watson and Francis Crick. These dudes found out the structure of this twisty thing. The more proper name for this twisty thing is double helix.
Each of the deena is made up of the nucleotides. These are the subunits of the DNA. Nucleotides are made up of a phosphate group, a 5-carbon sugar, and a nitrogenous base.
This 5-carbon sugar is called deoxyribose. Hence, DNA actually stands for deoxyribonucleic acid. The middle part of DNA is called the nitrogenous base. There are 4 different nitrogen bases. There is adenine, guanine, thymine, and cytosine. There are rules to these called base pairing rules, in which cytosine and guanine pair up like a car in a garage. Adenine pairs with thymine like apples into a tree. Now, any of these bases can be a pyrimidine or a purine. Adenine and guanine are purines while thymine and cytosine are pyrimidines. The outer part of the DNA is called the backbone. The backbone is made up of the sugar and the phosphate group. The double helix is held together by weak hydrogen bonds. Now, let's move on to step 2 on how to DNA. The replication of DNA is the ascendant part on how to DNA. The replication of DNA is called DNA replication. It occurs during the S-phase of interphase.
First, the DNA unwinds with the help of an enzyme called helicase. They do this by breaking down the hydrogen bonds. Proteins will then attach to each strand to keep them apart. Where the double helix separates is called the replication fork because of its Y shape.
Next, the DNA polymerases now add nucleotides to the exposed nitrogenous bases. Now we have formed 2 new double helixes. The polymerase finishes its job, and once it is done, the enzyme detaches. We now have 2 double helix made up of 1 old and 1 new strand.
There are many errors that are possible during DNA replication.
These are called point mutations. There are 3 types of point mutations: substitution, deletion, and insertion. Substitution is when one base is exchanged for another. Deletion is when DNA is lost or deleted. And insertion is when DNA has extra base pairs. There is also nonsense, missense, and frameshift. Nonsense is when the change in a base causes a cell to stop making proteins. Missense when a change of base pair results in a new protein. Frameshift is when loss of gain of DNA changes the DNA reading frame.
Though there is this slight mess up in the DNA, the polymerase comes in like kick-ass and fixes this problem. It does this by removing the incorrect nucleotide and replacing it with a good one.
The amoeba sisters are no matches for us. They will get the quick scope with the bomb on top. Like, subscribe, and blaze it to help us. Cell Brothers out.